That work? Great. Then now we can truly get started. I'm very excited to welcome you to the Dana-Farber TPD webinar, today run exceptionally in a hybrid format, as we have Georg, our very fantastic speaker, here with us at the Dana-Farber in Boston. And while Georg certainly needs no introduction to this audience in particular, let me just in case provide a few highlights. So Georg is a PI at the CeMM in Vienna, and his research group works at the interface of chemical biology, cancer, and gene control. And the Winter Lab pioneers high throughput and biased technologies to develop new strategies to both probe and disrupt aberrant transcription in cancer. And currently, their work largely focuses on proximity-inducing small molecules, including degraders. And Georg's contributions to the field of TPD were recognized with many awards, including the Tetrahedron Young Investigator Award, the European Federation of Medicinal Chemistry Prize, and the Appender Award. And last but not least, Georg is also a scientific co-founder of Proxygen and Solgate. So welcome back to the Dana-Farber. Thank you very much for visiting, and we're very excited for your talk. And to our Zoom audience, feel free to post the questions in the Q&A box, and we'll try to get to them at the end in the discussion.
Thank you, Susanna. Should we try to get that out of the way?
If you click more, you can hide it. Oh, yeah. Okay. There you go.
All right. Good. Thank you for the introduction. Welcome people here. Welcome people online. That's just my disclosure slide. Not so interesting. And I want to start by sharing something that probably is relatively self-evident to everyone, but the fact that target protein degradation is really dominated by these two modalities, heterobifunctional degraders on the one hand, PROTACs, and monovalent molecular glue degraders on the other side. They come with their known advantages and disadvantages. For PROTACs, the perspective of rational design, but then also the downside of that through their bifunctional approach and the thesis that you really need to engage the ligase and the target at the same time, you're also limited to ligandable targets. And through that building plan, they come with obvious PK or phys-chem limitations and a high associated molecular weight. On the other hand, molecular glue degraders typically only bind to the target or the ligase, and through that mechanism also enable the degradation of targets that are frequently considered undruggable or are unligandable. They're more drug-like through that nature of that molecular mechanism of action, but the big downside is that their discovery has been frequently driven by serendipity. And so I've worked in both of those fields, mostly on PROTACs during my postdoc time with Jay Bredner here at the Farber, and then mostly on molecular glue degraders in my independent research lab, where we really tried to innovate novel methodologies to enable a more rational discovery of molecular glue degraders at scale. I'm not going to talk about those ventures. I want to talk about two different kind of topics today. One, I want to share with you the perspective that by working in that field now for almost a decade, I've learned that there is actually more things that these two modalities have in common that might be separating them. And then lastly, I want to focus on targeted protein degradation outside these traditional proximity-inducing modalities. So to start with, I want to be very brief on a study that we have published earlier this year, because it has been presented in this audience already, where we got interested in this heterobifunctional PROTAC-like looking molecule, IBG1, that we found in the patent literature. Funny enough, actually invented by one of my former Bredner Lab colleagues who happens to work at Mitsubishi Pharmaceuticals. And there is a molecule that looks like a PROTAC. It has on the one side a ligand that binds to the base family of proteins, BD2, 3, and 4. And then on the other hand, it has this aryl-sulfonamide that we know binds to the E3 ligase DK15. And we got interested in that molecule because it is a very potent degrader of BD2, 3, and 4, particularly BD4, but that degradation activity of that molecule is independent of DK15. So in a cellular background where we knock out DK15, as you see in this immunoplot, the compound is just as active. And this was really the starting point for a collaboration between our group, spearheaded by Matthias, and then Alessia Juli's group, Oli and Engels. And so based on that data, we asked the obvious question, well, if it's not DK15 underpinning that molecular mechanism of action, what is the ligase that is actually hijacked by this compound? And we turned to a technology that we use very frequently in the lab, which are FACS-based CRISPR screens that allow us to ask the question, knockout of which genes, either genome-wide or focusing on E3 ligases, prevents the degradation activity of a given compound of interest. And by doing so, we identified very clearly an enrichment of another E3 ligase called DK16. And so you might fairly ask, this doesn't sound all that exciting, moving from DK15 to DK16. With that slide, I just want to share with you that there was no structural similarity that would have allowed us to say, well, if it's not 15, it has to be 16. So it really came out of the blue for us. We also were particularly interested by the observation that while our molecule binds to DK15, it has no binding affinity to DK16. So we're binding to the ligase that we don't need, but we're not binding to ligase that is functionally required. We also made the observation that in order for these molecules to induce potent BAD4 degradation, we need both of the N-terminal chromodomains of BAD4 in place, which is a very unique and distinctive feature of that molecule as opposed to any other heterobifunctional BAD4 degrader that we have. So there were many open question marks and open questions and question marks around all that data, and everything became much clearer when Angus solved the cryo-EM structure of this ternary assembly. And so what you see here is that this molecule, while bifunctional in nature, doesn't work like a PROTAC that would connect the target BAD4 and the ligase in trans. What happens is that this molecule binds to the target protein BAD4 twice in a cis configuration. So we know that the known BAD4 chromodomain ligand JK1 binds to, in this case, chromodomain 2, and in this aerosulfonamide warhead that has a known affinity for the ligase actually binds to the other chromodomain. And this arrangement of these two chromodomains is then sufficient to nucleate an interaction with the ligase DK16 that is mediated by several protein-protein interactions, but also small molecule protein interactions. And I don't have enough time to share with you additional experiments that we conducted, but I can tell you that based on that structure, we could show that ration improvement via structure-based drug discovery was feasible. We created very potent intramolecular glue degraders that work, I think, in the single-digit picomolar range. We could expand this to another ligase, DK11, and we learned that for both of those ligases, what is really important is that there's an intrinsic affinity between the target BAD4 and that ligase, typically in the single-digit micromolar range. And so we think that this PROTAC-like but glue-like, PROTAC-like looking but glue-like acting molecule is really a differentiated pharmacology that combines advantages of both the PROTAC and the molecular glue work. So far I've shown you a molecule that looks like a PROTAC but acts like a glue. Another I would say, example of why this PROTAC versus glue differentiation is not so easy, I want to illustrate via another story that we published earlier this year, where we initially wanted to develop a fragment or library of PROTAC-like molecules by connecting the well-known FKBP12 ligand SLF with different linkers to a set of covalent fragments, by creating a library of around 400 PROTAC-like candidates. And we wanted to basically hold novel E3 ligases in a function-first manner, inspired heavily by what Cravat did, for instance, and hoping that one of these covalent fragments would latch onto an E3 ligase with an induced proximity, and we would measure that in a cell-based assay in cells, leveraging cells that express FKBP12 nanoluciferase fusions. And so,
could you perhaps bring closer to the
yeah.
mic for the benefit of our online audience?
Yeah. I hope it's better now. So we ran that screen, and it was a beautiful looking result. So we have many compounds that didn't do anything. We had the cereblon-based positive controls that did something, and then we had one compound that had a very strong effect. And it was so far so good, but then despair set in when we realized that it was actually the negative control that had the strongest effect. And so it was then Chrysanthi who realized that while this initially looks like bad news, it is actually interesting because that structure of these alkylamine-based degraders was reminiscent of two previously disclosed compounds, one out of Genentech that had a similar chemical, let's call it degradation tail, appended to a ligand to the E3 ligase CyEP, inducing degradation. And there was another study out of Cheryl Aerosmith's lab reporting on an NSD2 degrader that had, again, shared very similar chemical features. So we realized that it's probably interesting enough to try to work up that mechanism. So the first question we asked is, what is the selectivity on a proteome-wide scale? From the expression proteomics together with Steve Gigi here at the Harvard Medical School, and realized that this molecule at the 1 micromolar is very, very potent. We see only FKBP12 destabilized or degraded upon 6-hour treatment. We could also show that in order for this molecule to work, the integrity of this primary amine is essential, because when we acetylate it, as you see to your right, that compound is completely inactive. So the question that emerged then, of course, again, is what is the ligase that drives that effect? And again, we turned to our FACS-based CRISPR screen, again, knocking out every ligase encoded in the human genome, asking knockout of which ligase prevents this molecule, sp3N, from inducing FKBP12 degradation. And we saw a very nice and very strong enrichment of a ligase called FBXL22. And I don't have enough time to share with you all the validation that we did in arrayed, FACS-based, or on Western blot. But typically, when we run these kind of screens and we see a 250-fold enrichment, we feel relatively good that this is going to validate. What I can share with you is an orthogonal assay where we said, well, if we functionally require that ligase, let's test whether this molecule can also induce cellular proximity between FKBP12 and FBXL22. So we treated cells, we set up a nanobit assay, and could clearly see that the sp3N, so the compound that has the free amine, can induce a proximity or in a time-ranging manner between these two proteins, while the acetylated degradation incompetent molecule is not able of doing the same. So we knew that these primary amines are metabolic liabilities. And there was a paper, a preprint published by David Nee and colleagues, again from Cheryl Aerosmith's lab, on this NSD2 molecule that hinted towards a potential metabolic conversion of these free amines that goes by amine oxidases that can be found, for instance, in FCS, and that convert the amine into a reactive aldehyde. So inspired by that work, we wanted to understand whether similarly our molecule would undergo such a metabolic conversion. And indeed, using target mass spec, we could determine that there is an accumulation of these reactive aldehyde species over time. We could show that indeed, in order for our molecule to induce degradation, we need FCS or FCS to be present. So in the absence of FCS, that compound is not able of inducing FKBP12 degradation. We could, however, rescue the dependency on FBS when we pre-treat the compound with recombinant amine oxidase, and so you see this here, minus DOA and plus DOA, so we can make the incapable, or we can render the amine into the aldehyde and then treat cells with that compound and reconstitute degradation. And when we finally synthesize the aldehyde from scratch, we can again see that this molecule is capable of inducing degradation also in the absence of FBS. So based on that, we surmise that the free amine is converted via amine oxidases that are found in FBS into the aldehyde. And the aldehyde is the reactive and active species that induces FKBP12 degradation. So aldehydes are known to interact with cysteines in a reversible covalent manner. So what Chrysanthi did next is that she mutated a suite of cysteine residues that are in the FBXL22 substrate binding pocket and identified one cysteine, which is 326, where when you mutate the cysteine to an alanine, she clearly sees complete rescue of degradation, so suggesting that in order for our compound to work, we really need that cysteine in place. We then could recapitulate this functional degradation assay with a proximity induction assay, again turning to the nanobit assay. So when you now treat cells with the aldehyde, we again see a very nice induction of proximity with wild-type FKBP12, but this induced proximity is abrogated when the cysteine is converted to an alanine. So now we could move to an in vitro setting, which was not possible before because we were working with the amine. So now we can work with the aldehyde, and now we teamed up with Brenda Schulman's lab, in particular Jakob Fuhrmann here, helping us with a recombinant ubiquitination assay, where again, what you can see here is that when we co-incubate FTP12 with wild-type FBXO22 and our compound of interest, we see a very nice FTP12 polyubiquitination, and this again is absent when we mutate this 1cysteine residue. We can nicely recapitulate the functional dependency on that cysteine residue that we observed in cells, also in a biochemical setup. And lastly, we could also show target engagement on that particular cysteine residue via intact mass spec. So here you see again either wild-type or mutant FBXO22 incubated with the aldehyde, and we see a nice adduction via this native MS approach at this particular cysteine. So next, we wanted to understand the reactivity or the selectivity of this molecule, and again, teamed up with Steve's lab, who performed activity-based proteomics profiling that allowed us to on the one hand, measure that we have around 30% target engagement of FBXO22, or that particular cysteine, and that I would say this is considering that there's around 20,000 cysteines quantified, is a relatively selective molecule with around 10 additional cysteines that we could that we showed are liganded with this particular molecule. So I mentioned earlier also that we got motivation to even work up the mechanism of this sp3N molecule because there were two other molecules reported. Here you now see the chemical structure, so they all share this alkylamine structure, but they do bind to different targets, so FTP12, CyEP, and NSD2. And so we wanted to understand whether this mechanism that we have worked out for FTP12 that depends on FBXO22 and that depends on the covalent adduction of this particular cysteine 326 is conserved or is generalizable also for these other targets. And the answer is yes. So here you see us knockout FBXO22 knockout cells that are reconstituted either with wild-type cDNA or again with a cysteine to alanine mutation. And you do see that the degradation that is elicited by this compound on the respective target is present in wild-type, but is abrogated as soon as we touch that cysteine residue. Letting us conclude that this is really a shared mechanism of action of how you can add such a alkylamine degradation tail to a ligand and kind of tapping into the same molecular recognition on that particular ligand. And again, I want to give a shout out to David Nee's paper that published a similar finding, very orthogonal strategy because they use proteomics to come to that conclusion with these NSD2 ligands, and both papers appeared relatively at equal times earlier this year. So my first task for today was to tell you that this differentiation between PROTACs and glues that we sometimes present as such a given fact is maybe not as obvious. And I tried to do that by first showing you a compound that looks like a PROTAC but acts like a glue, and then by showing you a compound that doesn't look like a PROTAC but still might act like one. And so I think it's really important that we keep an eye on those things that are common between these two modalities, which really is that they typically induce proximity between a target and a ligase. The ligase sometimes has a pre-existing affinity for the target, but it's typically not the ligase that turns over the target in a native environment. And both of those modalities also benefit from cooperativity and ternary complex stability. But I want to dedicate the rest of this presentation is really to think about degradation phenomena or degradation mechanisms that don't fit into this box of proximity induction. And I think there are some great examples, some of which were elucidated in Ben's and Eric's lab, of small molecules that were designed to be inhibitors but ended up inducing target protein degradation. We know this for BCL6, this is the example I just cited. We know it also for EZH2, we know it for estrogen receptor, we know it for menin inhibitors, and we predominantly also know it for kinases and kinase inhibitors, with also a lot of work from Lynn Jones along those lines, and I think I even cite one paper here. Yeah, good, well done. So I think if you mine the literature, there's many of those examples where we say, oh, this inhibitor induces the degradation of that particular target. And we try to find, is there a common logic, a common denominator? And I think there is one, and I think this is typically what all of these mechanisms or what all of these phenomena get ascribed to. And this is that most kinases, or a big chunk of all the kinases, depend on chaperones, on client proteins, particularly HSP90, for maintaining their structural integrity. And so once an ATP competitive inhibitor binds to a kinase, this communication between the kinase and the chaperone can be disrupted, and that can lead to basically the kinase losing its structural integrity, unfolding, and then being degraded in a cellular environment. So what we wanted to understand here are two things. We really wanted to quantify this phenomenon of inhibitor-induced target degradation in a systematic way, again, focusing on kinase and kinase inhibitors. And we wanted to understand whether there's some level of generalizable framework beyond chaperone deprivation. And so I'm going to tell you two stories. One is on 98 kinases, and one is on another protein called IDO1 that I'm going to come to towards the end of my talk. So we're going to start with the kinase project, and this is really the project of Natalie Scott, a postdoc in the lab, who set out a very brave effort to answer this question on a systematic level. And she had the idea that we can just take kinase cDNAs, express them in frame with luciferase, and then treat those cells with a kinase inhibitor of interest and track kinase abundance in time-ranging intervals. We just see what happens with a kinase abundance over time after treating with an inhibitor. And what Natalie did is she generated 100 cell lines that express 98 different kinase nanoluciferase fusions, or two controls, one being GFP nanolook control, which is a very stable protein, and one being a DGFP nanolook control, a very unstable protein, which kind of allows us to frame the data with respect to protein stability. And then we exposed these 100 cell lines to an annotated kinase inhibitor library of around 1,500 inhibitors. And for each of these inhibitor kinase or inhibitor cell line combinations, we recorded these time-ranging intervals. And so overall, I think she generated and analyzed around 1.5 million data points, and there's a lot of data to play around with. And this is exactly what we did. And we're working here with Patrick Aloy from IRB in Barcelona. And in very simple terms, what we're looking here are outlier effects. We want to see a kinase react different to exposure of a particular inhibitor of interest as opposed to the remaining 97 kinases, although as opposed to our two stability controls. And we're also framing, again, the effect of an inhibitor with regards to two controls, one being NBP2. This is a transcriptional inhibitor, so we block transcription elongation by inhibiting of CDK9, and then cyclohexamide, where we just block translation. What we want to see is a dose-result effect of the inhibitor that outperforms both of those controls. And so when we do that, we quantified 160 selective and around 300 semi-selective inhibitor-induced degradation events. What I mean by that should become clear here. So on your right, you see a scenario of a selective event, where it's really only this one kinase CDR1 that gets destabilized over time. What you see on your left is a trajectory of a semi-selective hit, where not all the kinases, but it's only 3 kinases that are affected and 95 kinases that are largely ineffective. So by recording all of the data, we can start trying to answer some of these questions on a holistic level. And the first is to, with this correlative evidence, understand how frequently chaperone deprivation is involved. And so we very clearly see that among those kinases that are destabilized, we see a very clear enrichment for kinases that are annotated either as strong or weak HSP90 clients. So I think this is an indirect yet compelling evidence that indeed chaperone deprivation is likely a mechanistic principle that underlies many of these events. Having said that, we also see many kinases that are destabilized that are annotated as known clients, and we also see cases of, and I'll come to that later, of known client kinases that are degraded, but where the mechanism is really not chaperone deprivation. So an example how that looks like, again, this is screening data, primary screening data on HER2, which is the most frequently degraded kinase in the kinase panel. Here, after cellular treatment with a compound neratinib. So what we can do here is we can now take all the hits that induce HER2 degradation, map them on a target landscape, and what we see here is that most of the hits really nicely cluster in a target space that would be represented by a known HER2 inhibitors. This is a long-winded way of explaining to you that many of the effects that we see here are likely on target and directly acting. It's something we could prove because many of those HER2 destabilizers are covalent in nature, so we could just mutate the cysteine in HER2 that we know is adducted by those inhibitors, and we saw in each of those scenarios rescue from degradation. We were at this point excited that maybe covalency is a feature that is more prone, or this could be that destabilizers are more prone in having covalent warheads, but this is something that did not necessarily hold true on a global scale. Again, two high-level analyses that were important for us. The first one is that frequency of degradation is not correlated to half-life. That was important for us to see because we're still worried that we're just quantifying transcriptional interference, but that doesn't seem to be the case. We also took the chance to compare likelihood of being degraded by an inhibitor and and likelihood of being degraded by a heterobifunctional degrader or PROTAC. Again, here using data from Catherine and Fleur, published in 2020. And what was interesting for us to see is that there's really no correlation. So we can use the degradation of kinases with inhibitors that are relatively PROTAC, and vice versa. There are kinases that are very easy to pursue with a PROTAC that we never see destabilized after inhibitor treatment. And we took this as another evidence that the kind of degradation that we observed here with these inhibitors is really differentiated from the typically or classically proximity-induced degraders that we studied. So out of these 160 examples, we had to pick some for mechanistic validation, and we picked 3, which I think is already a brave effort, given that most of the papers in our field work up the mechanism of one inhibitor or one degrader. But we thought it's important to share or to test 3 because that allowed us to also pick kinases that fall into different buckets in terms of their HSP90 client status. So we focused on mechanistic understanding of an inhibitor TAK285 that induces the destabilization of a TAK family kinase called BLK. A second one, so BLK is a weak client. A second one induces the degradation of the kinase LIN, which is a strong client. And then the third example is an inhibitor called RA4 that induces the degradation of RIF kinase 2, which is a non-client of HSP90. And again, due to time reasons, I can't go into detail for all 3 of them. We published that in a preprint earlier this year, so I invite you to go to the preprint and read up on all the mechanistic nuances. I'm going to focus mostly on RIF kinase 2 and the degradation that is elicited by RA4. So for all of our degradation work, the most important first check was for us that it's really degradation, so that the perturbation happens at the level of protein stability and is really independent of transcriptional interference. So again, we turn to these protein stability reporters where our protein of interest, here RIPK2, is expressed in frame with BFP separated by a P2A site from M-cherry. So again, we can always use these BFP M-cherry ratios to approximate compound-induced effect on protein stability. And what you see here in this fact plot is that we clearly affect protein stability. We can also do this again in time-ranging intervals, and this is a very nice degradation that we cannot quite recapitulate with an HSP90 inhibitor, as expected for that target. So again, then we turn to our workhorse, genome-wide CRISPR screen, asking again, what are the effectors that compound needs in order to induce RIPK2 degradation? What you see clearly here, this looks different from the other scalar volcano plots that I've shared with you before, where we see this very strong enrichment of one E3 ligase. In fact, there's not a single ligase that we saw enriched, but there were many proteins that we know are involved in membrane trafficking, and there's one protein that is called HEMO1 that has a ubiquitin-like domain. And many of those observations, if you look at them from a systematic perspective, would suggest that lysosomal degradation is in place, which is something we could validate. So when we pre-treat cells with, or co-treat cells with bacillomycin, can indeed induce a very nice rescue of degradation. And we can also see that when we knock out KIP200, which is a master regulator of macroautophagy, likewise, we do abrogate degradation of RIPK2 by this particular inhibitor. We then had a closer look to what happens to RIPK2 prior to degradation. Using microscopy, what you see, or we can hopefully see here, is that soon after ligand exposure, so look at the top row, RIPK2, prior to being degraded, forms these aggregates. And this is something, again, the clearance of these aggregates is blocked when we block or when we treat cells with papillomycin. So they're blocked via lysosomal degradation. And so going into the literature, what Natalie realized is that this formation of aggregates is something that is really part of RIPK2's biology. So in fact, RIPK2, when interacting with this pattern recognition receptor NOD1 that recognizes, for instance, bacterial species, peptidoglycans, what happens is that then these ribosomes, these structures formed, and they're called ribosomes. This is something that is dependent on the CAR domain of both RIPK2 and NOD1, which is something that we could also recapitulate. So also our inhibitors are dependent on the CAR domain. Suggesting, I don't have the slide to show that here. You just need to believe me, or you go to the preprint and read it out. So they suggested to us that whatever this inhibitor is doing, it's closely resembling the physiological state of how RIPK2 in this case gets activated and then cleared via lysosomal degradation. We also wanted to understand the role of TMUC1 in that process. And again, here using microscopy, what we can nicely see is that upon ligand exposure, TMUC1 co-localizes with these RIPK2 aggregates. And when we now knock out RIPK2, what you see, this is now the middle part compared to the top, middle row compared to the top row, is that the formation of these aggregates is clearly slowed down. So in the end, they still get these condensates, or these aggregates still get degraded, but TMUC1 apparently plays a role in the molecular recognition of those condensates, licensing them for lysosomal degradation. Again, this is something that we can also quantify in 2 different knockouts via Western blot. So the model that we have for this particular vignette is really that RIPK2 can live in 2 different states. One is the monomeric, that is stable, and one is the oligomeric, that is unstable and cleared by lysosomal degradation. And very similar to how these aggregates form upon physiological exposure, physiological regulation via NOD1, although our inhibitor can induce formation of structures that resemble these ribosomes, and they are then cleared by degradation. So what the inhibitor does is basically it tilts the balance between the active and the inactive state of these 2 proteins. The stable and the unstable state of these 2 proteins. Very briefly, the experiments we did with a second inhibitor, ASI3, where we see this very acute and very strong degradation of the kinase LIN. Again, here the screening data. What we did first is that we could prove that that inhibitor again directly binds to LIN in recombinant setup. We can again show that the loss of LIN that we measure in our screen occurs on protein stability level via our protein stability reporter. And what was very interesting for us is now here, we really needed to block both proteasomal and lysosomal degradation to rescue degradation. Apparently, whatever that inhibitor does, it pushes LIN through both of these cellular degradation pathways. So in addition to a CRISPR screen that I don't show here, what we also did is proximity biosimulation coupled to proteomics, asking which other E3 ligases are in the proximity of LIN when we treat with this particular inhibitor ASI3. And the answer is yes. We see 2 E3 ligases, CBL and CBL-B, that are very nicely enriched. Again, this is something we could validate here with, again, stability reporter. And similar to the redundancy that we see on the proteasome versus lysosomal degradation, also here we need to knock out both of those ligases to have a complete rescue. And again, what we realized at this point is that, again, CBL and CBL-B are both involved in the physiological degradation of hyperactive LIN. LIN can be hyperactive, for instance, after IgE stimulation. And what we then learned is that there is a kinase called CSK that really maintains LIN in this inactive state. And that prompted us to test whether our inhibitor that in recombinant assay we knew would be binding to LIN, maybe in cells also binds to and inhibits this kinase CSK. And to our surprise, we could not recapitulate in a cellular environment the target engagement of our inhibitor to LIN, but it was a very potent inhibitor of CSK. And alongside additional validation work that you find in the preprint, that again led us to conclude that we have here another scenario where the kinase lives in two different states, an active state that is unstable, and an inactive state that is stable. CSK is the master regulator, and by phosphorylating LIN, it triggers the inactive state. What we do with these inhibitors, an indirect effect, we block CSK and therefore move the needle towards the active, and in this case, unstable form of LIN. The last example is an inhibitor called TAK285, where we see the destabilization of the Src family kinase PLK, which is, as all Src family kinases, anchored to the membrane. So again, we could recapitulate. Perturbation happens at the level of protein stability. And so again, here we turn to FACS-based CRISPR screens. Here we can see a very nice enrichment of 4 genes, and interesting enough, these are 4 out of 4 subunits of the gamma secretase complex, transmembrane protease complex that so far I think no one has linked to targeted protein degradation. And what we realized, or what Natalie realized, is that what happens is that gamma secretase, upon treatment with or upon cellular exposure of this inhibitor, pushes the membrane-anchored BLK into a cytosolic form. And again, BLK here, when bound to the membrane, is stable. When cytosolic, is unstable. So it's another example of where we somehow tilt the balance with an inhibitor between 2 states of BLK, arresting it or moving it towards the unstable state. We don't know all the ins and outs of the molecular recognition of how the small molecule manages to reprogram or rewire the gamma secretase. We don't know exactly where gamma secretase cleaves on that kinase, but this is something that we're actively looking into in the moment. So as a summary, what we have learned from this systematic survey is that this inhibitor-induced kinase degradation is a frequently occurring event. We can clearly affirm chaperone deprivation as a broadly applicable mechanistic concept, but for those 3 examples where we had a closer look, we also have observed that what these inhibitors do is really supercharging physiologically relevant degradation pathways. So they are basically, in each of those examples, we have observed that the kinase can live in 2 different states, one that is stable and one that is unstable. And in all of those cases, the inhibitor just pushed the system towards the unstable state. Mechanistically, we learned that this can happen via different phenotypes. One is cellular localization, the case of BLK. One is kinase activity, in the case of LIN, and one is kinase aggregation, in the case of RIPK2. So the last vignette that I want to share with you, and I hope I'm somehow reasonable in time, is trying to extrapolate that concept from kinases to other enzymes or other protein targets. And this is a very nice collaboration we had with Herbert Wadmann's lab at the Max Planck in Dortmund. And the Wadmann lab is interested in phenotypic drug discovery. They were here particularly interested in a protein called IDO1, which is an interferon gamma-induced protein that is involved in tryptophan to kynurenine conversion. It's a heme-binding protein. It has been for quite some time been considered as a very hot target in immuno-oncology because this tryptophan kynurenine conversion is very interesting immunometabolic cue. So by setting up a phenotypic assay that has kynurenine levels as a readout, Herbert's lab identified these pseudo-natural products that they call IDEC because it's, what they realized is that it's a very potent inhibitor of the IDO1 enzymatic function, but it also leads to IDO1 destabilization, as shown here by Western blot. This destabilization is again very selective. So you see here on a proteome-wide scale, there's only really 2 proteins that are degraded, IDO1 and then DOCK8. Could show that there's cellular target engagement of these molecules and IDO1 using cellular thermal shift assays. And interestingly, they could also solve the crystal structure of these series of molecules, IDEX bound to IDO1. And what is, I think, remarkable here is that IDO1 does bind, or I think IDEX2 here binds to the heme binding pocket. And that's the same binding pocket that is leveraged also by clinically studied IDO1 inhibitors, such as linrotostat here. But what is very particular about IDEX is that binding to that heme binding pocket triggers this conformational dynamic at the C-terminus. So you see this C-terminal bundle of helices that are shifted, and there is particularly the last helix that where we lose complete resolution in the structure that is solved with IDEX as opposed to structure that is solved with linrotostat. So this structural flexibility that is induced upon ligand exposure gave us a first hint. This was also something that we could recapitulate with our stability reporters, because only when we put the BFP marker on the N-terminus, we could recapitulate the degradation activity of those compounds. As soon as we put BFP on the C-terminus and disrupt the potential putative molecular recognition that occurs at this terminus, those molecules were inefficacious. Again, we wanted to understand how these molecules are working, and we identified, again, a very strong enrichment for another culinary ligase, KLHDC3 here, a known C-androl ligase. We could show that this knockout of this ligase indeed rescues degradation activity of the IDEX, but we could also again show that they also stabilize IDO1, so they increase the baseline stability levels as shown by the bar graph here. This pointing us to, again, the fact that KLHDC3 will be involved in the physiological regulation of IDO1. We again teamed up here with Dennis Scott and Brenda Schulman's lab and set up an in vitro ubiquitination assay, where we asked the question, how potently is ApoIDO1, so IDO1 not bound to heme, ubiquitinated by KLHDC3. And what you see here when we treat ApoIDO1 with DMSO, so it's not heme bound, you see a very potent ubiquitination happening already after 7 seconds. If IDO1 is bound by heme, or if it's bound by linrotostat, that ubiquitination is markedly slowed down. However, that is not the case when it's incubated with IDEX6. So that suggests really that if APO1 is heme bound, it's less good of a substrate as opposed to its APO form, and somehow binding of IDEX6 to IDO1 mimics this APO state. We could again see similar trends on a cellular level. So when we perturb heme levels, you do see that IDO1 protein stability goes down, and this is again something that we can rescue when we supply cells or when we reconstitute heme levels. So leading us again with a final model where IDO1 is either apo or holo, so bound or non-bound to heme. When it's not bound to heme, it's very efficiently recognized and turned over by this ligase KLHDC3. When it's heme bound, that recognition is not as strong, and then it is in a more stable state. Again, when the IDEX binds to the heme binding pocket, they manage to mimic an apo state that is then more potently turned over. Again, kind of concluding on an effect where these IDEX again tilt the balance of protein stability towards an unstable state. So we have a fourth example that is not a kinase, but it's happening on IDO1. And in this case, we are not talking about activity, oligomerization, or localization with the kinase, but here's really modulating cofactor engagement, and then dependent on cofactor engagement, the protein IDO1 is either more or less stable. And so I want to conclude on that slide where I hope that I could convince you of two things. One, that these modalities that depend on proximity induction, while we tend to depict them as two separate entities, I would argue have more things in common than separating them. And that I hope I could also raise your interest in these inhibitor-based degradation events that I think really live outside of this traditional proximity induction world, will have very interesting molecular mechanisms that underpin them, and I think there's a lot of things that we can still learn on that field. I want to conclude by thanking the people in the lab that did the work. In chronological order, this was Matthias on the intramolecular bivalent glue story. This is Grisanti on the FBXL22 story, and Natalie both on the kinase and IDO1 story. I think all of our many collaboration partners, you've seen that we like to work together with people. I think it's more fun and it's much more productive. I want to thank funding sources. I want to thank people that showed up here and people online, and I'm happy to take any questions.